Good afternoon, students. Today we discuss about fermentation technology, that our unit 9. Now that fermentation, fermentation is in vitro process which is used for the production of or required product by using the microorganism. In a fermentation, this microorganism grow in a fermenter, that grow on a media in a fermenter, okay, and then add the raw materials. These microorganisms take a nutrient from the media, grow it, okay, and then utilize that raw materials, convert the raw material into the product. That process is called as a fermentation. The microorganism itself produces the some fermented product. Now the product which is to be produced by the process of fermentation, simply that wine product, that ethanol, then antibiotics, some hormones, some proteins, some enzymes. These are produced by the process of fermentation. Now in a fermentation, this process is carried out under the control conditions. The control condition is that the fermentation required that specific temperature is required, then specific that pH is required, some media is required, then all that oxygen, carbon dioxide is required. All that conditions are maintained and the fermentation process is carried out. Now for the process of fermentation, that fermenter is used. This fermenter which is to be used, nothing but the vessel. Then that vessels work under the control conditions. Now that vessels, that is nothing but fermenter which is to be used for the process of fermentation. Now having these are the diagram of the fermenter. Then this fermenter is made up of that either stainless steel or metal, which having provision for the inflow of fresh media and then outflow of the used media. Then fermenter also having provision for the entry of that sterile air. Then that sampling point is here. Sampling point means that microorganism which is to be grow on a media. This microorganism are transferred through this point in a fermenter. Then there is a impeller are present just for this impeller are used for the mixing of that media, mixing of that microorganism, okay, so that that oxygen is reaches to each and every part of that fermenter, each and every part of the microorganism. Now this fermenter which is to be used are control work under the control conditions. Now it is also called as a bioreactor. Now the difference between the fermenter and bioreactor, that chemical process which is to be carried out in a fermenter in a vessel, that is called as a fermenter. And now in a vessels that the bacteria is grow, by using the bacteria there is a production of that our required product. Now that is called as a bioreactor. Now this is the structure of that fermenter. Then that is there are some ideal properties of fermenter. Now first ideal property is that this fermenter must withstand the pressure of large volume of the liquid media, the media which is to be added in a fermenter. That media, the pressure of that media withstand by the fermenter, must be withstand by the fermenter. These are the first property. Second, this fermenter made up of that such a materials so that this fermenter that must not corrode. There is no corrosion of the fermenter, so that the materials which is to be used in a fermenter are the good. Then next is that in aerobic fermentation process, the oxygen must reaches to each and every part of the media, to each and every part of the fermenter. That's why again these are the properties of fermenter. Then the carbon dioxide, next property is a carbon dioxide which is to be produced in a fermentation process must be removed out from this fermenter. Again these are the provision. Next provision is that now fermenter work in a such a way that there is a stirring of the media. Because of the stirring of the media, there is an equal distribution of nutrient throughout the medium, okay, and then each and every bacteria take a nutrient and grow in a fermenter. Next is that in a fermenter, that is a foam is formed as the fermentation process proceed. During which stirring and aeration, this will be foam formation. This foam affect the product yield and also the growth of microbe. That's why the fermenter must control that foam formation. Next property is that during the growth of microorganism, fermenter must be maintained as predetermined temperature. That required temperature is maintained by the fermenter, which is to be required for the growth of bacteria. Then that fermenter should contain aseptic, means that fermenter must maintain the aseptic condition. Under aseptic condition the fermenter work. Under aseptic condition, that the specific bacteria which is or fungi which is work in that fermenter, that specific bacteria or fungi must aseptically enter in a fermenter. Next, now after completion of process, or that the used media must be removed from the fermenter, there is a provision. There must be drain at the bottom of the fermenter through which the complete fermentation broth can be removed. That's why this fermenter must having this provision, and the mechanism should be there for monitoring pH of the media. Okay, again there is a fermenter must provide that there is a provision to maintain the pH of the media. Okay, these are some ideal properties of fermenter. This fermenter must having these properties, and then that the fermenter which having this property, now it is called as the ideal fermenter. Now the working of fermenter, how this fermenter is work. Okay, you know that fermentation process. In a fermentation process, microorganisms grow on a media, and the fermenter work under the control condition at specific temperature, specific pH, inflow of the fresh media, outflow of the used media. Then this all that conditions are maintained. Then in a control conditions, that microorganism grow in a fermenter, and that raw materials convert into our required product. Okay, and now it is called as a fermented product. This is some working of the fermentation. Now then after completion of process, now that finally the product which is to be produced in a fermenter, that product are purified, isolated and purified. For that this downstream process is used. This downstream process is used for the isolation and purification of the final product which is to be produced in a fermenter. Now that is a downstream process. Next point is a downstream process. What is downstream process? This process is used for the after completion of fermentation process, the product is produced in a fermenter. Finally, that the product is purified, isolated and purified by using the different steps. That process is called as a downstream process. Now there are 5 different steps of downstream process: solid liquid interaction, release of intracellular product, concentration of the product, purification of the product by chromatography, and the formulation of the product, means that finally that the product is formulated. These are some steps which is to be involved in a fermentation. Now there are some outlines which is to be used by the downstream process. The outline is that first thing is that is a direct product in the culture broth. After completion of process, the product is produced. Now the product either produces inside the bacterial cell or either produces in a media. There are extracellular and then intracellular product. If the product is intracellular, it means that the product produces inside the bacterial cell. That's why now the next process is carried out, that is in a ferment cell disruption. It is necessary to remove out that the required product from the bacterial cell. That's why the first the cell is disrupt, break down. First the cell is isolated, then the cell is break down. By the process there are physical process, chemical process or enzymatic process or method is used for the cell disruption. Physical process means by using the mechanical pressure is applied, and because of that the cell are disrupted. In a chemical process, some chemicals are used for the disruption of the cell. In enzymatic methods, required enzymes are used for the breakdown of cell. These are the cell disruption. If the product is intracellular, if the product is extracellular, means that product produces inside the media. The product produces by the microorganism, and after that the product transfer in a media, that is extracellular product. That's why we does not require that bacterial cell. That's why the bacterial cells are removed and the media is removed. Okay, that's why broth with solid and liquid. Broth with, if the product is extracellular, the bacterial cells are removed and the broth is separated, because that the product is present in a broth in a media. Then the first step which is to be used, that is solid liquid separation. In a solid liquid separation, now that product is first separated from the media by using fluation, flocculation, filtration or centrifugation. Now the product is present in a broth, then by the process of either filtration or centrifugation, the product are filtered. By the process of centrifugation, the product are centrifuged and settle at the bottom. By the process of flocculation, product are floccules, formation of floccules or fluations. By using this, the product are separated from the media. Next is concentration. After separation of product from the broth, there is a concentration. Concentration means the product are concentrated. Now for that, the method which is to be used, that is evaporation, liquid extraction, membrane filtration, precipitation, adsorption. These different methods are used for the concentration of product, and the product are concentrated at one point, either at the bottom, either at the middle of the media, or either at the on a surface, at the surface of that media. Okay, and after concentration of product, the product are purified by using a chromatographic. Different chromatographic methods are used, either gel filtration, ion exchange chromatography, affinity chromatography, or hydrophobic interaction. These different methods are used. The methods of chromatography which is to be used for the purification of product are purified here, and after purification of the product, there is a formulation. Formulation, the drying, freeze drying or crystallization. These methods are used for the formulation of the product. Okay, means simply here in a formulation, the product are purified, and finally the adjuvants or any stabilizers or the preservative are added in a formula with that product. Okay, and finally that the product is formulated, and then this final product then use or transfer in a market. Okay, now this is that downstream process which is to be used for the purification of, for the separation of product from the fermenter after production of product in a fermenter. Okay, by the process of fermentation technology. Now this is about the fermentation technology. Thank you.